Hello everyone, today we are going to study about the simple and inhibition kinetics. Okay, so let's start this topic. In this topic, we will study about the introduction. Firstly, we see the introduction, then enzyme and their characteristic, and then the Michaelis-Menten mechanism or equation and the mechanism of enzyme inhibition. Okay, so let's start without any delay. Fine. So, firstly, we study about the enzyme and their characteristic. Okay, see, enzymes are the biological catalyst. We all know that enzymes are the biological catalyst and they speed up chemical reaction in the living organism. Enzymes are the biological catalyst that speed up reaction in living organism, often under mild physiological condition. Under the mild physiological condition. See, virtually all the reaction occurring within a cell that require the action of an enzyme, okay, as most of these reactions will not occur at an appreciable rate under the physiological conditions of the cell. So the term enzyme that was introduced by W. Curie in 1878 from a Greek word that is known as in. Okay, in it means in, inside, and zyme it means yeast, inside the yeast. Okay, that is referred to a certain substances like yeast and that made it cause a fermentation. Okay, so basically the enzymes are mostly protein. They are the mostly protein. When we are talking about the composition, they are the mostly protein, but some RNA molecule can act as enzyme like ribozymes. So the enzymes are the mostly protein. They are made up of protein like ribonucleic acids, molecules. They also act as an enzyme and they are found in all forms of life from bacteria to plants and the animals. Okay, so the fact that enzymes are protein that was established by the American biochemist that is known as James Sumner. Okay, James Sumner in the James Sumner. I am writing here. I think it's not visible due to camera. James Sumner in 1926. Okay, who crystallized Jake bean. What they crystallized? They discovered in the Jack bean urease the enzyme that catalyzed the cleavage of urea to ammonia and the carbon dioxide and showed it to be a protein. Fine. So the enzyme act by providing an alternate path of lower activation energy for a reaction to occur at much higher rates than those in their absence. Okay, fine. Now, like catalysts, they also increase the rate of reaction without involving themselves, okay, or without themselves being consumed or permanently altered by the reaction and without altering the chemical equilibrium between reactants and the product. Okay, so we can say that most of the enzymes are protein. These work at the specific temperature or within a narrow range of temperature. Okay, so at a higher temperature, the enzyme may get denatured and then enzymes lose their activity. Similarly, they also have maximum activity at the optimum pH. They also have a good activity at the optimum pH. The enzyme activity decreased at pH higher or lower than this. Fine. So the enzyme catalyzed reaction occur under the relatively mild condition, under the very mild, or you can say the mild condition. Fine. Now the temperature at which they work is the 100 degrees Celsius. Okay, below 100 degrees Celsius, atmospheric pressure, nearly you can say the neutral pH, whereas chemical catalysts that often require harsher conditions of temperature, pressure and pH. So further like that, catalysts, the enzymes are required in very small amount, to affect the large quantities of the reactant. Okay, so if the conditions are changed, and become unfavorable, then they can be poisoned. Okay, so now we discuss about their characteristic. So what are their characteristics? The important characteristics of the enzyme. So fine. They are the catalytic power, specificity and the regulation. They are the most important characteristics of the enzyme. Fine. So we study one by one the catalytic power. What are the catalytic power? The enzymes, see, the enzymes has a very high catalytic power. They have very high catalytic power. Okay, they have very high catalytic power and these may increase the rate of reaction by about, you can say that 10 to the power 8 to 10 to the power 12 times in that in their absence. The enzyme have very high catalytic power. These may increase the rate of reaction by the 10 power 8 to 10 power 12 times. Okay, and that is the rate of enzyme catalyzed reaction. That may be, you can say that several orders of magnitude or magnitude greater than those of the corresponding chemically catalyzed reaction. For example, we can say that a single molecule of the enzyme, a single molecule of the enzyme that is catalase. Okay, the catalase, catalase, catalase can catalyze the decomposition of about 10 million molecules of hydrogen peroxide. 10 million molecules of hydrogen peroxide. Okay, the single molecule